Yeah, participants, this is my pleasure to introduce myself. I'm Victorien Douillon. I'm a microbiologist at the University of Abomey-Calavi in Benin. And I will be with Dr. Boris Legba to talk about one of our study, which provides some lessons for improving the science policy society interfaces. This project was about improving the quality of laboratory diagnosis for bacteria causing bloodstream infection and better antimicrobial resistance control in Benin. And it was a real nice study done to improve the quality of the lab testing in Benin, and providing a lot of recommendation so that the stakeholder can use our basis to change the activities. And as you may know, the diagnosis of bloodstream infection in Africa, especially in Benin, is a real public health problem. And we need to reinforce the different level of intervention so that to get good impact on better results. And that's what we did during this project. We had, firstly, a gap analysis, which was conducted. And after that, we have this really nice the trainer approach that was used to train 50 laboratory staff involved in blood culture, antibiotic susceptibility testing, and quality assurance in bacteriology. And after that, we had many workshops to improve the dialogues between clinicians and laboratory experts, and also to share the project result with the authority of the Ministry of Health. And as a result, we compiled a set of recommendations for the improvement of the laboratory diagnosis of infection, the prescription and delivery of antibiotics, and optimizing the role of laboratory in the surveillance of bacterial resistance to antibiotics. And all those activities lead us to give many recommendations to the stakeholders. It's give us many lessons. It's help us to provide data to understand how deep the problem was and how this can be overcome. So I will let my colleague, Dr. the recommendation based on this study.
Thank you, Dr. Douillon. So after this study, we have proposed some recommendation which can improve the situation of laboratory diagnostic in Benin. And firstly, we can say that the reliability of laboratory results depends on the technical platform, but also of the skill of the staff. So we think that we need to prioritize the technical platform, but also organize some continuous training program for laboratory professionals, and be focused on quality assurance, because it is very important if you want to ensure accurate diagnosis and management of infection in Benin. We also think that it is very important and crucial to improve the collaboration between clinician, physician, but also other healthcare professionals, because it will be important to implement antimicrobial stewardship to optimize antibiotic prescription, but also dispensing. And we think that to achieve this, we need to recommend to the development of a national antibiotic therapy guideline based on AST results, antibiotic susceptibility testing, but also bacterial ecology. So at conclusion, we think that if you want to improve the policy science interface in the context of antimicrobial resistance, but also laboratory diagnostic in Benin, we need to prioritize the technical platform. We need to implement some continuous training for laboratory professional and also improve the collaboration between the health professional and the policy actor, because this will lead to have a better situation in Benin. So thank you for viewing our initiative. Bye.
Thank you very much, and nice session to all of you.
Bye.
Good morning or good afternoon, depending where you are. My name is Dr. Stephen Mofamadi. I'm going to be talking about harnessing the power of nanotechnology to achieve sustainable development goals in South Africa and beyond. If South Africa wanted to achieve some of the 17 sustainable goals, they need to find a technology that can assist them. One of the technology such can do that is nanotechnology. Nanotechnology is essentially a manipulation of matter on a nearly atomic scales, which is 1 to 100 nanometer, to produce the structure, material, and device. If you're looking at this slide, you can see some smaller thing that is not visible. This is how actually what happened when we're working on nanomaterials. We're talking about something smaller that is difficult to see with the naked eyes. But when we're using microscopy, such as electron microscopy, scanning microscopy, we're able to visualize those tiny particles that are smaller than 100 nanometer, even smaller than a virus. We're able to see their structure, their morphology, and also looking at their movement or their behavior. In healthcare or in SDG number 3, we see nanotechnology can improve diagnostic and treatment of infectious disease, neglected tropical disease, non-communicable disease, and in Nelson Mandela University, what we are working on, we're working on a nanodrugs and point-of-care device for cancer and diabetics, and we're also working on a green nanotechnology for antimicrobial resistance and artificial blood for blood transfusion. We are also trying to design or engineer the robotic nanotechnology-based air purifier that will play a role in terms of capturing, deactivating, neutralizing, inhibiting growth of microbacterial tuberculosis, influenza, coronavirus, and many other influenza viruses. And also they can inhibit this in the air or in the surface environment. We also believe South Africa wanted to achieve some of the SDG, particularly SDG number 6, where they need to use nanomaterial because nanomaterials can improve the detection, the treatment of water pollutant, and providing human with a pollution-free water. We at Nelson Mandela were trying to designing a nanosensor that can detect water contaminant, membrane that can do water purification, and green nanotechnology for water treatment. We believe this can play a role in terms of providing safe drinking water. We also believe that if South Africa wanted to achieve some of the challenges, particularly the SDG 1 and 2, they need to use nanotechnology because it can improve the efficacy of agrochemical soil integrity, resilience of agricultural systems while promoting food security. We know very well that nanosensors can also play a role in terms of precision farming, but particularly for those small farmers that are within the rural areas, they can be able to produce quality food. They can monitor the growth of the plant using such nanosensors and using fertilizer to provide quality products that come from these productions. Energy is one of the challenges in South Africa. We believe that South Africa, since they're moving out from fossil fuel to renewable energy, they can try to be ready. They can use nanotechnology to assist them to better the energy storage using such as nanotechnology. And also we believe that for all this to be realized, the impact of nanotechnology, we believe that the government and regulatory agency need to develop a clear regulating guideline for nanotechnology application and nanotechnology-enabled product to protect and to improve human health, safety, and environment. We also believe that in order to attract the private sector and business, the policymaker and government need to create a predictable, stable, effective regulating environment that encourages investment in nanotechnology innovation. We also believe that the government and policymaker need to set a code of conduct for nanotechnology researchers to ensure the safety of the staff, the students, the researchers, and community at large. We also believe that the government and the scientists, they need to increase public awareness and educate the public about the benefit and the risk of nanotechnology innovation, enable them to make informed decisions. And I thank you.
Genome editing is an innovative technology that is blooming in molecular biology, giving its precision, low cost, and ease of use. It is at the reach of every laboratory in the world, becoming the model of democratization in science. This technology generates DNA sequence variants at defined positions in the genome. It can be applied to modified coding regions affecting protein function, or in regulatory regions, impacting cell type specificity, time of promoter activity, as well as splicing variants. The best known system in genome editing toolbox is CRISPR-Cas9 platform. Alternative systems are TALEN and zinc finger nucleases. All these editing tools target specific DNA sequences in the genome, inducing DNA double-strand breaks at the target size. Once the DNA is cut, the cell uses its own machinery to repair, consisting in two major mechanisms: homologous directed repair and non-homologous end joining, resulting in targeted integration or gene disruption, respectively. The potential that genome editing offers to human health is immense. As a matter of fact, it provides more targeted treatments and improves the human condition through the repair of deleterious genetic mutations. Being a novel and sophisticated technology, it needs accurate regulation to ensure its use in a safe, effective, and most importantly, ethical manner. In plants, the technology offers unique opportunities for new agricultural transformation, providing new plant variants that hold improved crop yields, reduced levels of agrochemical, as well as improved nutritional values. In addition, climate change is affecting crop yields due to an increase in temperature, as well as creating water shortage. This technology surpasses conventional breeding systems, which are labor-intense and too slow to go from the first crosses to the commercial varieties. Likewise, genetically modified crops have been generated with beneficial traits through the transfer of transgenes or gene elements of known function. Despite the success, certain GMOs are still affected by unproven food security and environmental safety concerns from the general public. To this respect, the potential and negligible risks involved in modifying genomes through the genome editing technology are significantly lower than those of GMOs, because most modification consists of only few nucleotides, producing changes similar to those naturally occurring in normal breedings. Therefore, the use of genome editing into modern breeding programs can modernize plant breeding via rapid, cheap, and precise crop improvements, as well as meeting the ever-increase of global demand for food and water supply. In addition, government regulation and consumer acceptance are likely to be more welcoming towards the use of this new breeding technology.
I am Emanuele Buratti, Group Leader of Molecular Pathology at ICGB. Very briefly, what I will do in the next 2 minutes is to tell you what is the advantages of having omics technologies in human health. So basically, by omics technologies, what we mean is a set of experimental procedures that are mostly automated and which allow to understand all the global changes to our DNA, our RNAs, and our proteins in our body following the onset of complex pathologies. And by complex pathologies, I mean, for example, cancer, for example, neurodegeneration, but also viral infections. And basically, what these omics technologies allow to do is they allow to give scientists a very deep view in all the changes that occur in our body following a pathological condition. And this will have very important consequences. First of all, when we will try to set up personalized treatments based on the way that each of our individual bodies responds to disease or viral infection, but also in clinical trials, it will allow to stratify patients in order to understand if for any given treatment there is one specific type of patient which responds better to a type of treatment, and therefore be able to measure a lot more efficiently the efficacy of new drugs that have come to the market.
My name is Vittorio Venturi. I'm a group leader at the ICGB, the International Center for Genetic Engineering and Biotechnology, located in Trieste in Italy. So I would like to discuss with you briefly how the omics sciences are important in plant biotechnology. So plant scientists today are interested to understand how plants tolerate to abiotic stresses like high temperature, water scarcity, or high saline conditions, or how they cope to resist a pathogen upon, how when a pathogen attacks a plant, how come certain plants are resistant. So we need to understand these mechanisms of resistance in order then to transfer them to economically important plants. So the genomics allows us to sequence the genomes of plants. The transcriptomics allows us to understand which genes are transcribed under these stressful conditions. The proteomics allows us to have a complete picture of all the proteins and their abundance present in plant cells under these stresses. And the metabolomics allow us to understand which are the small metabolites, small compounds that are abundant under these stressful conditions. So this gives us a lot of information and will allow us to understand these resistance to these stresses and how we can then use these mechanisms to create economically resistant plant varieties. I also wish to mention that these omics technologies are and will be very important in the study of plant microbiomes, because we now know that plants, just like humans, live together with many microbes, and this microbial community is very important for plant health. So these omics, this genomics, transcriptomics, proteomics, and metabolomics will allow us to study the microbial communities and how they live together with the plant, and how which types of microbes and what they're doing together with plants, and what kind of communication, what kind of signaling, what kind of metabolic activities they have. So then we can use this information also to devise new ways, sustainable ways for the next generation agriculture.
Hi everyone, my name is Maria Cecilia San Martin, and I will be presenting you this brief report rewritten with Dr. Gabriela Bortz from Argentina, titled Between Regional Convergence and Locally Rooted Technological and Cultural Aspects. Lessons from the case of COVID vaccines in Latin America. In developing countries where the pandemic struck in context of limited resources and inequalities, the health challenge was even more critical, including different key aspects such as accentuation of the center's semi-periphery dependency in terms of limited vaccine access, global context of concentration of vaccine development and manufacturing in the global north, limited resources and inequalities, and a public scrutiny of the role of the state. In this global context of concentration of vaccine development and manufacturing and the jurisdictional expansion of intellectual property, several middle-income countries ventured into different initiatives to increase their access to vaccines. On the one hand, some of them requested temporary exemption of any type of IP on medical products and the associated technologies related to the prevention, diagnosis, and treatment of COVID. On the other hand, some developing countries opt to their own vaccine production. Regarding some facts about the pandemic responses in Latin American region, the pandemic marked a repositioning of the role of the state and the scientific and technological sector, mostly public. The latter was summoned as a key actor and authoritative voice, hoping to contribute to the resolution of social problems. In a context of historical disconnection between science and technological capacities and socioproductive needs, the health emergency gave rise to a flexible articulation between the state, the scientific and technological sector, and the private sectors in Latin America, that enabled to obtain useful knowledge and technologies that to respond to the pandemic at an unprecedented speed. This was possible due to the strategic and oriented policies deployed by the states to address economic, financial, technological, regulatory, and IP issues, to greater institutional integration in terms of scientific and technological planning and financing and regulatory aspects, and to a change in the research and development and production dynamics of biopharmaceutical laboratories, both public and private. In the Argentinian case, the state focused initially on the acquisition of foreign vaccines, both through its own negotiation and the COVAX mechanism, and then focused on the local production of vaccines designed and developed abroad. Simultaneously, both the public, scientific and technological sector and the private biopharmaceutical laboratories endeavored on the design and development of national COVID vaccines. In the case of Brazil, amidst a lack of coordinated health response at the national level, the country opted for acquiring foreign vaccines. And in a second phase, the government of São Paulo and the Ministry of Health signed agreements with foreign pharmaceutical companies to transfer vaccine technologies for domestic manufacturing. Here we provide some public policy recommendations oriented to promote access to vaccines, taking into account some factors including particular institutional dynamics, forms of governance and coalitions between key actors. 1. Focus on local specificities. There's a need for public policies that encourage promoting the sector in the long term, with convergence between different areas. A key aspect for large-scale biopharmaceutical projects. These policies must take into account local and regional economic, political, social, and cultural specificities in order to aspire to intra-regional collaboration. The search for regional convergence through regional value chains requires the implementation and coordination of public policies that promote regional value change, as well as regional IP and health regulation harmonization. 3. Regulatory issues. The strong demand for vaccine access in pursuit of the public interest in an emergency context conflicted with the traditional approval processes for biologics deployed by regulatory agencies, which require rigorous scientific evidence of safety and efficacy. It also conflicted with local root imaginaries, past memories of state failures and power abuses, which must be taken into account when formulating public policies regarding regulatory issues and health technologies. 4. Role of the scientific and technological sector. The development of local vaccines, such as the Arvac Cecilia Grierson in Argentina and Butanvac in Brazil, show the change in a public-private interaction dynamics, from more linear to more articulated. In the Argentinian case, the designs of the vaccine took into account the available private manufacturing capacities and the fact that it would be administered in booster vaccination campaigns. As a local development, the researchers imprinted into the technology design imaginaries of technology and health sovereignty, as well as they took into account the future conditions of distribution of the vaccine when selecting the vaccine technology. In the Brazilian case, previous public sector capacities acquired in the local production of influenza vaccine were mobilized in order to speed up the production and approval process of the Butanvac vaccine. 5. Developing complementary capabilities. Finally, the implementation of public policies at the regional level on complementary capacity building would allow for greater autonomy with respect to key biopharmaceutical input, as well as critical step towards the consolidation of regional hubs and value chains. Thank you very much.
This policy brief is on the alignment of the pharmaceutical industry with the Sustainable Development Goals, meaning pharmaceutical industry, the industry focused on the development, production, and commercialization of treatment for human diseases. So first, the pharmaceutical industry today is not sustainable. It emits more greenhouse gases than the automotive sector. And this is mainly due to the fact that it heavily relies on fossil oil for most of its industrial processes. Second, it's not equitable. Most of its investment in research and development is focused on diseases with a big prevalence in industrialized countries, such as cancer and mental disorders. And it has overlooked diseases which are a burden in most of the world population which live in developing countries, such as infectious diseases and neonatal conditions. So how to fix this? First, redefining the role of governments in innovation. Here, the rationale is that the pharmaceutical industry will be reluctant to take the lead of these initiatives because they will most likely mean a reduction on their financial benefits. So for that, governments will need resources. So these resources will come from a fairer share of the benefits of the successes in biomedical innovation. So with these resources, governments will run drug development programs focused on societal and not monetary benefit. Second, implement prevention and early detection programs, which has also been overlooked by the pharmaceutical industry, given that they represent slim financial benefits. Second, governments should promote drug repurposing and licensing of shelved drugs. So here the rationale is that by doing so, drugs that has been going through the development process will be translated into cheaper drugs, maximizing the use of the resources. So for that, novel IP policies will have to be created to make the protection of these drugs easier. Second, create initiatives like the Orphan Drug Act in the US to stimulate the participation of the industry in these initiatives. Third, stimulate the out-licensing of shelved drugs. And fourth, governments should create and lead an ecosystem focused on repurposed and shelved drugs. Third, governments should focus on natural products for the identification of new drugs. So here the rationale is that natural products will be a cleaner source of new drugs, and these drugs will be produced at an industrial scale, most likely by green chemistry. So for that, governments should develop novel IP policies to make the protection of these new drugs easier, to simplify the Nagoya Protocol. So that will also allow the countries where these natural products will come from, which are typically poor countries, to protect their biodiversity and use it as a source of economical activity. And third, once again, create an ecosystem focused on natural products, and again, the government being on the lead of this initiative. So what are the main challenges that these initiatives will face? First, redefining the role of governments. So governments will have to be on the lead on these initiatives in collaboration with the industry. So governments will have to imagine, execute, and finance these initiatives. And to make this sustainable in time, they will have to make sure to get a fair share of the successes. Third, government-industry partnerships will have to be redefined. So they will have to happen in a more equitable, constructive, and transparent way. Third, industry, as said before, will be reluctant to participate in these initiatives, but they will be needed for this, given their expertise and their access to technologies. So for that, in the industry, there will have to be a change of culture and mindset, which is, from our perspective, the biggest challenge of all.
In our paper, One Health, Breaking Institutional Silos and Achieving Health for All, 4PAUSE shares why we believe that the latest One Health definition can help governments tackle global challenges like antimicrobial resistance more effectively. And we will share policy recommendations on how to enable this development. The new iteration of One Health, which was developed by the One Health High-Level Expert Panel and endorsed by the Quadripartite, was operationalized as well within the Quadripartite One Health Joint Plan of Action. It frames One Health as an integrated, unifying approach which aims to sustainably optimize the health of people, animals, and ecosystems. It follows a whole-of-government, whole-of-society approach, meaning the right governmental institutions and the affected communities are involved in finding ways to tackle the interdisciplinary challenges. It is also rooted in prevention at source and tackling the root causes before an outbreak emerges. So it does not only examine how health risks in animals and the environment affect humans, but goes a step further to address the ways in which human activity affects animals and the environment, thereby driving disease outbreaks. Antimicrobial resistance is a global challenge that has been addressed through a global action plan on AMR, which guided national One Health action plans. And while both global action plans and national plans include important measures such as phasing out antimicrobials as growth promoters in animal farming and encouraging prudent use, the plans do not go far enough on engaging all the relevant sectors and reducing the need for antimicrobials to begin with. Given that more than 70% of the total amount of antimicrobials used globally end up in animal farming, tackling the problem at the root by improving animal welfare could bring about meaningful impact. This is a key area to focus on because poor animal welfare causes chronic stress, which leads to weakened immunity in animals, leaving them more susceptible to infections and resulting in increased use of antimicrobials. Poor management, stress-related behavior in animals, and animal husbandry, which includes breeding farm animals for high-performance traits, increases the prevalence of illness and injuries in animals, which may become infected, again calling for antimicrobial use. Experts have concluded that improving animal welfare reduces the risk of injury and infection in farm animals, and that rethinking production systems to ensure high welfare is essential for limiting the need for antimicrobials in farming. Such changes can be best enabled via a One Health approach that tackles the challenges at the root and brings to the table the affected communities as well as institutions across the human, animal, and environment interface. To enable health for all, animals, humans, and the environment, and based on our analysis, 4PAWS recommends science and knowledge-driven decision-making and policy development formalized in a digital interface for knowledge exchange, One Health coordination to break institutional silos and strengthen collaboration amongst relevant stakeholders and enable joint strategy development. One Health strategies beyond antimicrobial resistance and in line with the One Health Joint Plan of Action with measures across the human, animal, and environment sectors, which should be tied to preventing the drivers of outbreaks while ensuring that the communities at the front line of health challenges are part of the solution and supported to transition away from high-risk practices. Finally, countries with limited capabilities will need support in the development and implementation of One Health strategies by the institutions of the quadripartite and funding, which will be essential to ensure that resources are available for the implementation of national action plans. Thank you.
Hello, I'm Akila Ashraf, and today I would like to talk on a very important topic, that is impact of gender inequality and social stratification on antimicrobial resistance. My other group members are Wasim Ashraf and Peter Thompson. First of all, the introduction. Antimicrobial resistance is the potential in bacteria to persist and grow as a response to specific concentration of antimicrobial agent, and it has become a global threat worldwide and is in the limelight for past few years because of increasing trend in antimicrobial resistance. And there are people who are saying that this is going to be our next pandemic if we pay no attention to the uh, to the to this crucial issue of antimicrobial resistance, because we are the process of development of antimicrobial resistance is very fast, and it is we're going to end up in the days where no treatment is available for the diseases which were treatable in the past days. That is all because of the increasing trend of antimicrobial resistance. The rationale of the study was to assess the impact of gender differences and other socio-economic factors on antimicrobial resistance in Pakistan, and to find out how men and women in different age groups are differently at risk of antimicrobial resistance. The range of health programs and services are not equally available, accessible, affordable, and acceptable to all groups within the population. So it is widely acknowledged that a gender equity focus in all aspects is required to protect and improve population health. Before we go into further, it is important for us to understand the factors which are contributing towards the increasing trend of antimicrobial resistance. The known factors are mainly the excessive inappropriate and non-judicious use of antibiotics in healthcare, veterinary, and agricultural sectors. A pilot study has been conducted to actually check the effect of gender inequality on antimicrobial resistance, and to develop the foundation for the research on a given subject. It was mainly to assess association between patients' antimicrobial resistance status and several explanatory factors, namely gender, age, marital status, number of family members, education level, and employment status. The study has been divided into two parts. One is the lab work, in which the antimicrobial resistance profile was checked for the specimen obtained from various patients from the local community. And the second part is to fill in the questionnaire regarding all these questions, regarding all these factors mentioned in the slide, like gender, age, and educational level, etc. And the results we obtained are not sufficient to claim anything, because the number of subjects are not very high in the study, as it is a pilot study. But yes, there is found some significant differences in terms of effect of antimicrobial resistance in different age groups, in different people with different educational level and employment status. The reason behind them has to be worked out. The future recommendations based on this pilot study mainly include finding current evidence and identify priority linkages related to gender and antimicrobial resistance research. It is extremely important to find out. The second is to development of guidance on how to strengthen the gender inclusiveness of antimicrobial resistance interventions and implementations research projects in low middle income countries. Thirdly, the determination of key drivers is of highly importance for key drivers of antimicrobial resistance, with special focus on gender equity, income, education, age, and other gender related differences. And finally, to assess the differences in behavior of men and women towards use and misuse of antibiotics. So, to sum up all this, it is extremely important to find out how men and women and other people living in the society are at risk of development of antimicrobial resistance. To conclude this study, understanding of the effect of social stratification on AMR in the population studied will be used to improve the surveillance and to make recommendations for changes in policy for control of antimicrobial resistance. If you would like to contact me, my email address is mentioned in the slide below. Thank you very much for listening to me.